Bismillahirrahmanirrahim. We are starting our first lecture, and in the previous video I have told you that I'll proceed my lectures according to step 1 book. So the section first discussed in the step 1 book is molecular biochemistry. I've divided molecular biochemistry into the 5 sections. The first one is DNA fundamentals, then we'll discuss about purine and pyrimidine synthesis, then we'll discuss transcription, then translation, and finally DNA mutations and DNA repair. Let's talk about DNA fundamentals first, and for that purpose you have to move with me towards the board. You can see that there's a cell, and inside the cell there's a nucleus, and inside the nucleus there are chromosomes. Let me take one of the chromosome outside, and you can see that this chromosome, like the DNA, is highly tightened and compact form here. So this tightened and compact formed, when you see through the electron microscope, this portion is known as heterochromatin. Heterochromatin. This heterochromatin means highly condensed portion, right? A highly condensed portion, you can memorize it, taking H as highly and C of the chromatin as condensed, right? Highly condensed, which means that cell is not dividing at this stage, cell is not producing protein in this stage, right? So when you will see in a more open way, you'll find that the double helix loops twice around a protein, around an octamer protein that is known as histone proteins, right? This is histone protein. This is an octamer and having overall positive charge. Due to the lysine and arginine residues, there's a positive charge on the histone proteins, right? And DNA has overall negative charge. Overall negative charge is due to the phosphate group attached to the ribose sugar. So this negative charge is being attracted by the positive charge of histone protein. Now you can see that it loops around the histone protein, right? Histone protein. And this is the expressed portion of the DNA. This expressed portion of the DNA is known as euchromatin. Euchromatin. Euchromatin means expressed portion of DNA, means it is expressed for transcription. A certain type of protein is being transcribed by this expressed portion. So you can take this E for expressed, right? So there are two important terminologies, heterochromatin and euchromatin. When you see through the electron microscope, if you see the condensed portion, the highly tightened portion, you call it as heterochromatin. And if you see something that is expressed and that is opened up, that is euchromatin, right? Very clear? There is another type of protein that basically does what? It staples the DNA with the histone protein, and this is known as H1 linker. H1 linker proteins, right? It does what? It basically staples DNA with the histone proteins. So these are the very beginning of these two processes, and let's move toward an important process that is known as DNA methylation. DNA methylation. I hope everything is clear. DNA methylation is what? Here is a DNA, and you can see that there are nucleotides, and you know there are two types of nucleotides, purine and pyrimidine. We're going to discuss later on. And always, you know, adenine is linked with thymine, and guanine is linked with cytosine. Cytosine is linked with guanine, you can see. So almost 20% of the DNA is composed of CG sequence, right? So what happens actually here in DNA methylation, a methyl group is added to the cytosine. Methyl group is added to cytosine with the help of protein named as DNA methyl transferase, right? There are certain types of DNA methyl transferases that attach the methyl group to the cytosine. When this methyl group is added to the cytosine, there comes a protein that binds with methylated cytosine, right? This methylated cytosine binding proteins, methylated cytosine binding protein, MCBP. What does these methylated cytosine binding protein does? These proteins don't let this portion of DNA to be transcribed. So will make it what? The DNA methylation helps in the condensation. I told you that in condensed form, DNA is not producing any proteins, and to produce proteins, DNA has to transcribe. So the DNA methylation main function, the major function is to make the DNA mute. So we can memorize it like DNA methylation makes the DNA mute, M of the methylation for the mute purposes, right? It is a critical epigenetic process. DNA methylation is a critical epigenetic process. I can make a separate lecture over it, but the necessary information that is required for step 1 USMLE, we'll discuss only those. So what does this whole mechanism can determine? Let's discuss here. The first thing is aging. Aging, right? You know, aging is an inevitable process, and with the growing ages, there are certain type of proteins that are necessary in your childhood are not required in your when you grow older, right? So aging can be determined by the DNA methylation process. With the aging, more portion of the DNA goes into the heterochromatin form, and with this type of heterochromatin portion, you can determine, oh, this is the particular age of the person. So aging is determined by DNA methylation. The second process is carcinogenesis. Carcinogenesis, you know that cells divide rapidly, and there is a higher nuclear to cellular ratio. So what happens in the cancerous cells, more portion of the DNA is heterochromatin, is not transcribing, right? So there is a hypermethylation. Hypermethylation is in the carcinogenesis. So as I told you that methylation done by the DNA methyl transferases, so the drugs that inhibit that enzymes, right? Drug inhibiting this enzyme, DNA methyl transferases, won't let the cytosine to become methylated, more methylated. So you can prevent hypermethylation by giving certain drugs, that is anti-cancer drugs that acts on this enzyme and won't let cytosine to be methylated, to prevent the hypermethylation. This is the one way to control. But carcinogenesis, I told you that this is determined by the DNA methylation. Next come an important process, that is genomic imprinting. Genomic imprinting. What happens in genomic imprinting? You know very well that there are, let's suppose we'll take XX, right? There are two alleles of same gene on the two chromosomes. One, for example, let's say this is dominant and showing the characters, right? This portion is showing the character, and this is not showing the character. So the one who is showing the character is in the phase known as euchromatin phase, and the one that is not showing the characters is in the phase that is heterochromatin. For that purpose, to become heterochromatin, I told you that very important process is required, that is DNA methylation. So there will be hypermethylation to this portion, so that in the next progeny, it will be easily to determine that the characteristic will be shown by this chromosome, right? So this is genomic imprinting, silencing that portion that is not producing proteins. So genomic imprinting, in genomic imprinting, DNA methylation has an important role. The next one is, let me erase this one for the sake of convenience, an important process, that is leonization. Leonization. Leonization is, you can say, a female process, as there are two X chromosomes having the same characteristics, showing the same characteristics. So what happens actually, if this chromosome is expressing, this is not required to show the expression, having the same characteristics, having the same type of gene. So what happens, this becomes methylated, right? These chromosomes become methylated. So silencing one of the chromosome and ensuring that the next cell will show the proper, the genetic expression, this process making a silent one pair of X chromosome is known as leonization. So in the leonization, how it becomes silent? Because of hypermethylation, DNA methylation. So DNA methylation, for the methylation, you have to memorize that DNA methylation makes the DNA mute, right? So let's move toward another important process, that is histone methylation. Histone methylation, yes. So what happens in histone methylation? Let's discuss here first. Histone methylation, as I've drawn for the sake of convenience and for saving your time. This is histone and this is methylated. The two processes happen in the histone methylation. Histone methylation basically does what? It suppresses the reversible transcription, right? Reversible transcription we are going to discuss in the transcription section, but reversible transcription is prevented by the histone methylation, and sometimes it mutes the DNA too. So it does two processes. Histone methylation, histone methylation is making reversible transcription suppression, and also does what? The same process as like DNA methylation. So we can use a terminology that histone methylation mostly makes DNA mute. Let's discuss another thing, that is histone acetylation, right? Acetylation. If acetyl group is added to the histone, I told you that there is overall positive charge on the histone protein. So the acetylation of histone reduces its positivity. When the positivity of the histone will be reduced, right? So it will not attract the negative portion of DNA anymore. So DNA will be expressed. So for the expression or for the DNA to become euchromatin, histone acetylation is required. You can say DNA methylation and histone acetylation are antagonistic in their function. If DNA methylation is making heterochromatin, histone acetylation is required for euchromatin. So whenever cell is producing proteins, it's becoming expressive, histone acetylation is required. So histone acetylation means increased transcription, and DNA methylation means decreased transcription, making DNA more condensed, more compact, more tightened. And histone acetylation is making what? DNA more expressive, right? And increasing transcription. There is another important function, that is histone deacetylation. Is simply inverse of histone acetylation, means removal of acetyl group, right? Removal of acetyl group will again gives back its positivity and it will attract. So in histone deacetylation, decreased transcription, making more condensation, right? Condensation. So you can say DNA methylation and histone deacetylation, they both are condensing the DNA, are tightening the DNA, right? Making the heterochromatin. What? DNA methylation and histone deacetylation. And histone acetylation is required for the expressive DNA to transcribe and to translate into the proteins, right? Increases the transcription. So this is the process. These are the things, these are the basic fundamentals that are very necessary to be in your mind before going into the details of the DNA. So let's move toward further details